Hi, citizen scientists. I'm back, and today I want to highlight um, the importance of very, very small organisms that are part of the plankton that we rarely see in our microscopes because they're so small. And they're called nanoflagellates. So if you look at this picture that I'm showing you here, what you see is a cell of one of those L. polys, Lingulodinium polyedra, that we've been observing for the past few days. These are the bioluminescent organisms that have been causing the beautiful glowing at night in the waters of Southern California. But what I want to highlight here is the existence of those tiny, tiny little dots that you see swimming around this cell. So this cell is dying. This is a sample that I kept in a container that was completely sealed, so there was no oxygen, and it was also in the dark, so the phytoplankton could not even photosynthesize to produce oxygen for themselves. And so what you're seeing here is the process of decay of these L. poly cells. But as the sample gets older and it's, you know, dying and decomposing, you can see that now there are a number of very small organisms that we see here as these tiny little dots swimming around the L. poly. You see that? We can barely see their shapes, and I'm playing with the focus here a lot so that you can see, because they're moving really quickly in and out of focus. So right now, if you're looking at the L. poly cell, you're going to see a lot of tiny little dots swimming around it and also underneath it and even inside. So these nanoflagellates, they are heterotrophs, so they don't photosynthesize. They will be consuming the cells that are dying, and just like bacteria will do, but these are not bacteria. The bacteria are not going to be visible to us in this kind of microscope that I'm using. So I'm using this picture that you're seeing right now is magnified at 400 times what a normal human's eye would be able to see. Human eye, sorry. So we wouldn't be able to see any bacteria, but these are what we call, what I call the cleaning crew. So these tiny little nanoflagellates that will come in there, they range in size from about 2 to 20 micrometers, whatever you prefer to call the unit. So these organisms are very small, but still visible with a light microscope like the one that we have here. I'm going to try to move around and see if we can find another cell that's being eaten by nanoflagellates. So here you see a dot moving around this cell. You see the tiny little dots. Sometimes you can barely see their shade. As they move in a drop of water. There's about 1 ml of water here right now in my chamber, in my slide, which is a special slide that I use that fits 1 to 2 milliliters of water. All right, but that's it. As you can see, because there, do you see the tiny little little lines moving around? Not quite sure what those are, but they're big enough to be noticeable in my microscope. So they're not bacteria, but they're definitely part of this cleanup crew, as I call. So they'll come and they'll start consuming the dead cells, and that's part of this process of decay that blooms will go through. Now, obviously, this is a decay that was provoked by the fact that I placed a sample in a container and closed the lid, so I kind of forced the death process to occur more rapidly than what is actually happening in the ocean right now. But I just wanted to make sure you can see these organisms that came with the seawater, the nanoflagellates and all these organisms that are now consuming the dead little poly. They came with the seawater. They were not introduced by me or by my sampling procedure. So this is definitely part of the natural process that would take place in the ocean. Here's a big one here. So you can see there's a whole community that is interconnected and interdependent, and they all live together in these bodies of water, whether it's the ocean or a wetland or a bay or whatever the water is, traveling together in a current, for example. You would have the big, large dinoflagellates, the diatoms, and also this whole part of the plankton community that we rarely acknowledge, but they're so important. And in terms of biogeochemistry, they're also very important. They're also going to be affecting the balance of not only oxygen and CO2, but also the balances of other chemicals in the water, such as nitrogen and phosphorus. So pretty cool, right? Right. I hope you're enjoying these videos. I'm trying to make them a little less shaky, but hopefully if you are a Flow Citizen Scientist and now unable to come to the visitor center for our weekly meetings there, hopefully you are at least enjoying these virtual explorations of seawater samples and plankton samples. And if you're not a Flow Citizen Scientist, make sure to contact us. And if you're interested in phytoplankton ecology, contact us to learn more about our program, and you can join us in our weekly meetings to talk plankton. All right, that's it. Bye, everybody.